Today, we have Dr. Anish Mitra with us, which is a great pleasure for me because he's my friend. And he's an MD, PhD psychiatry resident here and neuroscientist. So I'll give a brief bio on him, and then I'll try to give a little bit of context about why we're talking about the default mode network as part of a psychedelic science group. And sorry, I'll just be admitting people as I talk, so excuse the interruptions. But so really briefly, Dr. Mitra is he originally studied math at Stanford, and he went on to do an MD PhD at Washington University in St. Louis, and he studied with Dr. Marcus Raichle, who was the first person to name and I think also discover this default mode network activity in the brain. Actually, looks like he's in the waiting room right now. I'm glad I got that part over with before I butchered the history. And then after finishing his PhD, he now came to the Stanford Psychiatry residency, where he is now a PGY2 resident. So really briefly, why are we talking about the default mode network? So first of all, there's a lot of literature about its relation to different states and disorders and functions in the realm of psychiatry. So its activity has been correlated with changes in quote unquote normal default mode activity have been seen in depression and anxiety and ADHD and PTSD and borderline personality disorder. So we see these various changes. We know it's a very important network. I'm going to let Anish talk about exactly what it is and how we define it before I reveal my ignorance on this. But why psychedelics? Around 2010, Robert Carhart Harris and Carl Friston out of Imperial College London essentially tried to marry Freudian ideas with these ideas about brain networks. And the proposal was that the Freudian idea of the ego was instantiated by this default mode. And he talked about this difference between a primary process, which is free and unbound, like dreaming or the childlike state, or REM sleep or the psychotic state, versus this secondary process or this bound or constrained mode of functioning or perceiving or this internal state, which he connected to the ego. So this was a proposal, and there was evidence for it that they suggested. And a couple years later, with some neuroimaging work with LSD and psilocybin, it was shown that actually LSD and psilocybin seem to correlate with a decrease in activity in metabolism and connectivity of this default mode. And I'll just pause to say hello, Dr. Raichle. I'm just giving a brief spiel here. I'm far underqualified to do this. That's why I'm going to turn this over quickly to Anish, but just finishing up this piece about why psychedelics and default mode network, but just hello. And so there's now, since 2012, I think was the first neuroimaging study, there have been a sequence of neuroimaging studies with different groups correlating LSD and psilocybin with essentially a reduced intrinsic functional connectivity of this default mode. And this has then been correlated with this idea of ego dissolution. So if the ego is instantiated by this default mode network, and when you ask people, they describe this sense of ego dissolution or oceanic boundlessness, that maybe those are paired. Maybe that is what's happening. But then recently there's been suggestion that this may not be specific to LSD and psilocybin, because we also see a similar pattern of reduced default mode network activity with MDMA and with salvia. Salvia not even messing with the serotonin system at all. Actually hitting opioid receptors. And then also with even things like alcohol and cannabis. And so the question is this effect, this reduction of activity, specific to the psychedelics? Is it really correlated well with this idea of ego dissolution? Is it crucial to the therapeutic benefit we see in some of these psychedelic trials? And the reason I'm so excited to have Anish speak in 15 seconds is because for a person who's not trained in statistics and functional connectivity and neuroimaging, we're sort of left guessing and left only with our own metaphors and the metaphors given to us. And the problem is the metaphors often mix with physics. And so when we talk about free energy and entropy and connectivity, we certainly understand them, or I certainly understand them on a metaphorical and sort of everyday use level, but I don't understand them on a physics level. Anish does, which is amazing to me, and I'm thrilled to have him. And I will now stop the stream of consciousness and turn it over to Dr. Anish Mitra for about 30 minutes, and then we'll have Q&A, and then Dr. Rekel, obviously, feel free to pop in at whatever point.
Awesome, thanks, Gianni. Man, I hope we all have the experience of having a hype man as amazing as Gianni opened for us. Like, I've never felt better in my life. And hey, Mark, I don't know that we've had a chance to chat yet this year, so Happy New Year, man. Well before the COVID-19 pandemic, Mark and I have succumbed to the pandemic of procrastination, so I asked Gianni to buy me some time while I finish these slides. So needless to say, I haven't had a chance to show Mark yet, so please interrupt me, Mark, as I get things wrong. And of course, you guys, this is very, very casual, so feel free to interrupt me throughout. I have about 30 minutes of talk, but I have a hard time keeping track of you know, the Zoom audience in addition to my talk. So I guess Yell and somebody will, whether myself or Gianni will.
I'll handle that stuff. And
cool.
if people just can post questions in the chat too, and I'll keep track.
Awesome. Can you guys see my screen right now?
Yep.
Awesome. Okay, so I want to talk about today demystifying the brain's default mode network, and I'll tell you a little bit about how this talk came to be. I was on the, let me see. Of course, here we go. So I was on the consult liaison service, you know, part of a resident. And the first week, earlier this year of that service, I was asked to give a talk. They asked all the residents to give a talk on Fridays, 15 minutes, whatever we want to talk. And I said, hey, I'd kind of like to talk about the default mode network because it comes up in psychiatry so much. And this is the response I got. My attending, we can talk about default mode network and other networks some other time, followed by an emoji that I don't really understand. My fellow, yes, hard pass on DMN, clapping, excited for HIV psych tomorrow, kindly suggesting what she thought I should be talking about. And so I pushed back slightly, mentioning that I was actually a PhD with Mark Rekel. So we were gonna go ahead and talk about the default mode network. So at the risk of referencing an early pandemic meme, I think it's safe to say that personally, I created like an early version of a primer on what the default mode network is from my perspective. And I think it's fair to say out of Mark Rekel Lab, our perspective. And I was talking about that with Gianni, and he was interested in it. So thanks, Gianni, for having that interest and inviting me to talk about it. So even though I feel like the default mode network is associated with Zen and like meditation, for me, the motivation came from a very un-Zen place, I think it's safe to say. But I'm sympathetic to sort of what this attending and this resident were getting at. So this is a figure that we made in the lab way back in 2015. It's hard to believe it was that long ago when Mark was writing a review for the annual review of neuroscience, if I remember right, talking about the default mode network. So what this figure represents is the first paper where Mark kind of described the default mode network in those words was published in 2001. And I think at the time that we were writing this, Mark, you might remember better than me, there were like 15 or 20,000 references to that paper, citations.
Not quite that many.
Okay.
It was in the thousands. It's roughly, I guess about 18,000 right now.
Okay. So safe to say it's been cited a lot. And so we just kind of wanted to get a sense for what's the work that kind of sprung out of it. And so we, you know, just made our hand at kind of a network analysis of who was citing who. And you can see that there's these kind of families of papers. So like there are these papers that are kind of task driven, trying to find tasks that increase activity in the default mode network. There's a lot of this kind of blue cluster was looking at the default mode network and disease states. Functional connectivity, of course, is its own literature. There's this kind of self-referential processing and mind wandering, which are not terms we necessarily love, but there's a huge literature on that. And then kind of neurophysiology and cell biology. And these were terms just pulled from NAP. But I think sometimes the frustration with the default mode network can be that it can be invoked to explain so many different things. And so that's why I kind of wanted to approach this as a demystifying thing. Like, what do we really understand about the science here? Because I think that on its own is really exciting. So my talk is not going to be exhaustive by any means. I just want to give a perspective on 3 questions. First, what is the default mode network? Second, what's the function of the default mode network? And then with an eye towards the purpose of Yanni's class, I'd like to kind of explore what role the default mode network might play. So I think it bears kind of going through the history of how the default mode was discovered. And that history involves PET imaging. And I'm kicking myself because the original PET machine that was a very homebrew WashU creation was like on my way into the office every day. And I don't have a picture of that, but I have so I just found this one on Google. And it's like a modern view of a PET machine. And I'm sure most of you are familiar, but just to repeat, what PET does, right, is it picks up radioactivity emitted by a tracer that's injected into your body. So you get some kind of tracer for whether it's, deoxyglucose, or it can be for blood flow, for oxygen. You inject that into the body and it goes into various places, and then you look at the radiation emitted by that tracer to where it binds or where it's taken up. If you use something like fluorodeoxyglucose, which is what this picture is, it turns out that a lot of glucose goes to your brain. And so you get this nice picture of where glucose is being utilized in the brain, or at least where it went. And that, is an inference about where energy is being used by the brain. And if you think about the brain using energy, as a first assumption, you can say that represents brain activity. That's something in the sodium ATPases that's helping to, move the ionic gradients for neurons to fire. But in terms of what you're actually seeing, it's a tracer of some other element, whether it's energy or something else. Now, I'm sure you guys have all seen lots of kind of nice images of parts of the brain lighting up in PET as a result of particular tasks. But I think it bears kind of repeating, or at least emphasizing, that when you look at the raw data, there's barely any differences between when you're engaged in the task that myself, the experimenter, wants you to be engaged in versus when you're doing barely anything in the scanner. So this is from a Nature paper that was the first paper to kind of show the human language network using functional imaging in humans. And here's the underlying data. This is humans sitting in the scanner, PET scanner, just staring at a little X. Here they're looking at nouns. Here they're reading those nouns out loud. And in this last condition, they're seeing a noun like ocean, and they're being asked to generate a verb that has something to do with that noun. So you might say surf, you can surf the ocean. And at least to me, if I saw these data come hot off the presses, I'd say, oh, it was a failure, right? Because I really don't see any differences. But then, and one of the reasons that's true, right, is that the brain is an enormously metabolically expensive organ all the time. Whether you're kind of zoned out listening to me drone on about the default mode network or kind of engaged in mental chess, the brain's metabolism is about 20% of your basal metabolism all the time. Now, luckily, Mark didn't give up as easily as I would have. And it turns out that when you do a statistical comparison of these conditions, you do see differences, right? So if you look at passively viewing words versus just staring at an X, you see some activity in parts of the visual network and parts of the network that are known to kind of be involved in language processing. Similarly, if you read those words out loud as compared to just kind of looking at them, you start to see kind of parts of the language network and parts of the auditory and motor networks become more involved. And finally, when you do this, really language task, where you're thinking about the language and generating a verb corresponding to a noun, you get this beautiful image of, Broca's and Wernicke's area. But these differences are small, right? There are a few percent in the uptake of, in this case, it was changes in blood flow, which is a longer story, but correspond to changes in neural activity. So there are changes, but they're small. Now, all of these numbers are positive, right? So when we're basically contrasting doing something a little bit more difficult with doing something a little bit easier in each of these conditions. And we have these positive numbers showing that some parts of the brain had slightly higher blood flow, and we're inferring slightly higher activity as a result of doing the more difficult thing. But the question I think was kind of lingering in people's minds is, what about the negative signs in these images? Are there parts of your brain that are actually less active when us as the experimenters are asking you to do the more difficult thing? What about the negative signs? And so this is a picture of basically those negative signs. And there's actually work before this, before the 2001 paper that kind of talks about this, but I think Mark really pulled it together in this manuscript where he showed, look, basically independent of what task we as an experimenter ask you to do, there are all these parts of the brain, massive parts of the brain, right? And in fancy places like the medial prefrontal cortex, the retrospenial cortex, the parietal lobe, that actually decrease their activity when you tune in and pay attention, as compared to when you're zoned out staring at an X or doing nothing at all. So these are all negative signs, all decreases in activity. And in this original paper, they looked at 9 different tasks, you know, the language task we just talked about, memory tasks, several visual search tasks, and basically didn't matter. Whenever we asked you to do something difficult that took your attention and pulled you out into the external world, this set of brain areas reduced their activity. So it's pretty invariant. And that's actually pretty interesting, right? Because the story before this was that there are specific parts of the brain that light up when you do something, right? So even though these differences are modest, you can see like what we talked about before, these topographies were different. So the fact that it's these same parts of the brain that are kind of diminishing their activity across this wide host of tasks was pretty interesting. So this, just to like come up with a very basic definition, is the definition of the default mode network, at least as it was originally conceived. It's this invariant set of parts of the brain that decrease their activity across a host of tasks, predominantly ones that kind of pull you to pay attention to something and not be introspective. So we can kind of go through these parts of the brain, right, and talk about them individually. So there's a whole lot of medial prefrontal cortex, and on the basis of other pieces of evidence, both like lesion studies from stroke, and also from some studies that specifically design tasks to increase activity in these areas. And I do want to be clear about that. It's not that every single task you could ever imagine causes these areas to decrease in blood flow and activity. It's just the vast majority do. But through some clever work, they found like, okay, the dorsal prefrontal cortex has some kind of evaluation of your actions and plans. Ventral medial prefrontal cortex, you know, has been long associated with reward stimulus associations. The nucleus accumbens shows up in this part of the in this network, and that's associated with reward signals. Hippocampus, of course, has a long and storied history in episodic memory, kind of the events of your life. You know, the parietal lobes is kind of interesting, right? Because they seem to be associated with space. So we all know about neglect from right parietal strokes. But there's also the left parietal area, which kind of people kind of associate with the cognitive space, like how different words, for example, like oranges and apples belong in like fruit space and things like that. And then finally, the kind of retrosplenial cortex, which really speaks, you know, it's strongly connected to the hippocampus and has been thought to kind of produce your map of the world, both in terms of the physical world around you, but also kind of your experiences and how you navigate around them in time. So I'll talk a little bit more about that later. But just like a explanation of the set of regions that kind of comprise the default mode network, how they were originally. Okay, so it's this constellation of regions that are more active in most kind of less cognitively demanding conditions as opposed to more cognitively demanding conditions, especially things that kind of pull you out of introspection and into the outside world. And of course, like I said, tasks can be designed to increase activity in some of these areas, which alongside lesion studies to kind of tell us something about the regions that are involved. So the kind of second, I think, wave of kind of studies that led to some work on the default mode network came through Fmri. And again, I think this is very familiar to you guys, but just to cover it in brief, Fmri depends on basically a magnetic resonance signal, right? And it happens to be an accident of nature that deoxyhemoglobin is paramagnetic. which means that when exposed to a magnetic field, it'll get lined up. And when that magnetic field is changed in particular ways, you can get a signal out of that from the electrons no longer being lined up. Now, what's important, right, is that, as I kind of mentioned before, and it's kind of a topic in its own right, is that blood flow increases when the brain becomes more active. The implication of that is that the ratio of deoxy to oxyhemoglobin changes. It turns out that the brain brings in more oxygen than it actually needs when it's active. And so you get much more oxygenated hemoglobin in the brain when it's active compared to deoxyhemoglobin. And that produces fMRI time series. And so these are just two examples of fMRI time series from two parts of the brain. It happens to be from the retrospenial cortex and the medial prefrontal cortex, part of the default mode network. And you can see that fMRI time series are slow, right? These are not action potentials. They're not at millisecond precision. But it's like kind of the slow activity over, minutes. And you can see here that the red and the blue lines are correlated. They seem to kind of go up and down together. That turned out to be a pretty revolutionary finding when it was first described by Bharat Biswal. Because up until this point, we had kind of been doing neuroscience using the PET regime, right? Where you do something slightly more difficult and slightly more easy, you subtract those two images, and that's how you try to find networks in the brain. So you can do that with fMRI, and this is what was done here in panel A, where people were asked to first sit in a scanner doing nothing, and then sit in the scanner while they were squeezing two balls in their hands. And sure enough, if you subtract those two images, you can kind of see the motor cortex because they were engaging their hands. What Bharat Biswal then showed is that if you look at that condition where the people were doing nothing at all and just ask, in reference to these time series here in A, so picking a couple of time series from a couple of parts of the brain, what's correlated with them? What goes up and down with them together, even when you're doing nothing at all? And it turns out you get a very similar image. And so what this established is that for some of the major classical networks in the brain, such as the motor cortex, their activity is coordinated and nearly synchronized, although not exactly synchronized, even when you're not engaging that network actively, at least from an experimental's perspective. So this became one of the properties of known networks. And you can see this in the visual network and other kind of classically defined networks in the brain. But the default mode network was somewhat controversial at this point. And so the question kind of naturally arose, does the default mode network have this property? Are its constituents more or less synchronized from an fMRI perspective? And so it was actually Mike Grecius here at Stanford who showed in 2008 that sure enough they are. So they took various parts, those constituents that we talked about of the default mode network, took their fMRI time series and just asked, are they correlated? Did they go up and down together? And the answer was yes. So that kind of created a whole nother field. This is kind of the field of functional connectivity. And whenever you hear functional connectivity, I just want you to think correlation. I heard a great joke once that the only difference between a correlation and a functional connectivity is what gets funded by the NIH. It's just literally time series that are correlated. But they do allow you to make these maps what the different networks of the brain are. The default mode network is one of them. Now, once in a while, you'll hear somebody kind of talk about, well, the default mode network is like the seed of humanity. It's what makes us special. I don't hear that much anymore, but suffice to say, I don't think that's the case because every mammalian species that we've looked at has a default mode network as defined by the property of this constellation of networks that go up and down in their activity together in fMRI. It's been found in rats, been found in monkeys, in humans, of course, also in mice, ferrets, and marmosets. And again, this is fascinating, right? Because not all the parts of the brain are homologous. Like you can see that the parietal areas here in the monkey are different than the parietal areas here in the human. But the basic, for example, retrospenial parietal kind of medial prefrontal parts of this are present in a monkey as they are in a human. And we could kind of trace that back to a rat, to other rodents, ferrets, and marmosets. So this seems to be a system that, at least in mammals, is quite conserved. This is a slight detour, but I just want to tip my hat to the fact that some amazing work has been done in terms of the genetics and the kind of individual cell connectivity in the default mode network. So this is a figure I actually pulled also from Mike Grecius's lab, published in 2015, where they used gene expression data. Ask, okay, these parts of the brain kind of change their activity together, but are they genetically more similar to each other than other parts of the brain? And sure enough, what they found is that that's true. So the parts in red here, we're looking at the medial prefrontal, the retrosplenial, and then some of the medial temporal parts of the brain here. They belong to the default mode network, and their gene expression and kind of the cells that they were pulling from those areas are more similar to each other than they are to other parts of the brain, the motor cortex here. So there's clearly some properties of these parts of the brain that they both work together, but they probably have these gene expression properties that distinguish them from other parts of the brain. There was also a really fascinating paper recently from the Allen Institute in Neuron here in 2020. And I couldn't find a picture of what I thought was the most interesting point, but they did just amazing kind of technical work. So they took mice, used Fmri to map out the default mode network as defined by correlations or functional connectivity. And then they did like individual viral tracing, the parts of the default mode network to particular cells to see how they were kind of connected. And they showed that cells within the default mode network are more connected to each other than they are to cells in other parts of the brain. And one kind of fascinating thing that, again, I couldn't find a great picture of was the retrospenial cortex, that part of the back middle here in humans, was found to kind of have the most connection to the default mode network, at least from using the tracers that they use, which is going to come up as something interesting here on. So returning to kind of the bigger systems fMRI work, the other kind of, I think, important fact that arose about the default mode network is that it seems to be one of basically two very large, very broad networks in the brain. And so like, for example, here, if you look at the traces in red and orange, you're looking at traces from kind of the retrosplenial and medial prefrontal areas, parts of the default mode network. And here in blue, you're looking at a part of the inferior parietal sulcus, which is part of something called an attention network. It tends to become more active when you're pulled out of kind of introspection and when you're pulled to pay attention to the outside world. If you look at the way that the hot and cold traces here relate to each other, you can see they're anti-correlated. When one goes up, the other goes down, and vice versa. And it turns out that this is a basic principle, that there tends to be at all times this kind of push-pull between, the colors are reversed here now, so I'm sorry it's a little confusing, but the elements of the default mode network, when they're kind of more active, these attention networks that pay attention to the outside world are a little bit less active, and vice versa. So I'm going to gloss over a lot of kind of research that's been done, but one hypothesis about what this means functionally is that you have kind of an egocentric and allocentric view of the world. So to start with the allocentric view, right, when you're kind of really engaged externally, you're paying attention to what's around you. It's less about how those things work in your own individual model of the world or how they relate to you, but how they relate to each other. So in this case, like there's just a bicycle and a bucket and I guess a windmill out in the world, and you're like really paying attention to like, where are these things? How can I find them? That kind of thing. But their second kind of view of the world is more egocentric, right? So in terms, like forgetting about their spatial location, what do they mean to you, right? So you could like imagine yourself in bed in the morning planning a beach day when you're like, okay, I want to go build sandcastles. So I'm going to grab my bucket, hop on the bike, and head to the pier where the windmill is, and that's where I'm going to spend my day. That's much of a more coming from your kind of internal model of the world and an egocentric kind of plan. And so the thought being that the default mode network makes things like this happen, whereas the kind of external, like where are these things, what do they mean, and like a veridical view of like where are these things in the actual world comes more from this allocentric model, which is sustained by the attention networks of the brain. So just to summarize this piece, the fMRI evidence shows that the elements of the default mode network are basically correlated in their activity. And this property is present in basically all mammalian species. And there are these really cool gene expression and cellular signatures that I think are portal to future amazing work. And they've kind of shown a special importance of the retrosplenial cortex and being connected to the rest of the default mode network. And then, glossing over a lot of work, it seems that the function in part of the default mode network is to kind of maintain this internal egocentric model of the external world. So with that, I'm going to just transition into the psychedelics part of what we might think about the default mode network and how they relate to psychedelic experiences. And actually, I'm going to start in a place that might seem kind of odd. I'm going to start by talking about Alzheimer's disease. So, we've talked about the parts of the brain, this is probably looking familiar by now, that are implicated in being a part of the default mode network. And here in the middle, you're looking at work that Randy Buckner did, mapping out using PET imaging where amyloid plaques in Alzheimer's disease accumulate. And I'm sure you're making the same leap that he made, which is to say, these two maps look pretty darn similar. Like the places where amyloid show up in the brain seems to map up pretty well into the parts of the brain that are part of the default mode network. And there's kind of more to the circumstantial story. If you look at where there's atrophy in the brain of people with default mode network, with Alzheimer's disease, it's in that same distribution. And similarly, if you look at where there's disruption of metabolism, you see the same kind of thing. So why is this important? I think we all have some level of experience with folks with Alzheimer's dementia. And clearly, you can see some of the hallmarks throughout the disease of how it affects your sense of self and your model of the world, right? So like one of the first things that happens in this disease is you'll often see patients who get lost in familiar space. Like they'll describe their experience of like not being able to make their way home along their commute. And then they'll have these word finding difficulties, right? And you guys remember how I mentioned that part of that parietal lobe is kind of helping to map out cognitive space and lexical space. How do words relate to each other? And so you can see potentially, right, how in Alzheimer's, you're seeing a natural experiment of how early degradation of the default mode network or a compromise in its function kind of leads to this kind of unfamiliarity or model of the world of things that are deeply familiar to you not working quite right. And then, of course, as the disease becomes more insidious and progressive, you do lose things like memory, but also you start to misinterpret things, right? You hear patients or perhaps the patient's granddaughter being mistaken for the patient's daughter. These kind of mistakes where, again, the experiences that you're having are, of course, being lined up with your brain's kind of expectations, but there's a mismatch there, right? Like you're making the wrong inferences. And again, I think there's a correlate there between some of what happens under a psychedelic experience. Of course, it's not nearly so profoundly scary, but in a sense, you do start to misinterpret signals in the veridical environment when you have a psychedelic experience. And certainly, I don't know the literature well enough. I'd love to be corrected on this, whether people can get lost in familiar places. But I've definitely seen in the literature that people kind of see familiar places with new eyes, right? Suddenly it doesn't seem so familiar. You start to notice things you never noticed before. So I think that, in a strange way, Alzheimer's has something to say about how this particular network and its function can kind of be correlated with what happens in a psychedelic experience. Now, I want to talk about one specific paper that came out of the Diceroth lab with two amazing first authors, Isaac Kuvar and Sam Basuna, friends of mine. And they used ketamine, which is often used at anesthetic doses as a dissociative anesthetic. So you could do surgery on folks under ketamine. But I don't know if you guys have had the experience of seeing people go into surgery on ketamine, but it is bizarre because their eyes are open and they're just staring at you. And one of the advantages of ketamine is that it does not depress your vital signs. So your heart rate's ticking along, you're breathing, and it's very disconcerting because they don't really look asleep. And actually the theory is that they're not truly anesthetized in terms of an arousal perspective, but they're profoundly dissociated. And at lower doses, ketamine has been well documented to produce elements of psychedelic experiences. So intense alterations in moods, your perception, you don't perceive what's around you. You can have like that kind of internal guided experience, body awareness changes. And people do tend to have this sense of not being in touch with their body. And often they'll experience ego dissolution as well. And I just pulled this from a paper and that was kind of funny. Some patients find these experiences terrifying, whereas others do not. So really ranging the gamut. So my friends in the lab did this lovely study where they were looking at the mouse brain here under ketamine. And, you know, we could start by looking at when you just give the mouse saline, and you can kind of see, you know, there's just changes in activity, kind of these wave-like patterns kind of propagating through, but nothing, at least for me, really draws my eye, right? But if you look at the mouse under ketamine, some of that stuff is still happening, but I'm going to play this again, but really keep your eye on this part of the brain. It's almost like a sine wave. It's just like flashing on and off at a very regular rhythm. It happens to be between 1 and 3 hertz. And it happens to be that this is the retrospenial cortex, one of the really the key areas of the default mode network. And in this view of the brain, which is taken using calcium imaging, the only part of the default mode network we can see. And so they did a lot of lovely work kind of proving that these mice were not fully anesthetized. They were still awake, they were able to move around, but they had these kind of correlates of dissociation, and as far as we can measure it in a mouse, they were kind of less kind of keyed into their body sensation. They didn't quite feel pain the same way. They were definitely a little bit more confused in familiar environments. So I think to a first approximation, these mice were having a psychedelic-like experience, not fully anesthetized. And the big change we see is that there's just this sine wave activity, this flashing activity in part of the default mode network, the retrospenial cortex. Now, so they did a lot of lovely work characterizing this, but the final piece of this paper was they actually found a patient here at Stanford, who I believe was first identified by Joseph Parvizi, a neurologist here. And this patient had seizures with aura that looked a lot like a psychedelic experience. He describes his auras, and so we can kind of look at this. He says, I was listening to 2 parts of my brain speak to each other in a way that the third part of my brain, which I considered to be me, was able to listen. So really kind of his ego kind of retreating from the rest of his brain. More experiences. What would it feel like if someone else were to come into your head? What I considered me shrank to this other part of me, where the other parts of my brain that were talking, I considered them me. Where in this 3D space am I? I took a blanket, I threw it over my body just to see, because I knew that when I don't feel it, I don't consider it me, and immediately my legs were no longer a part of me. So this is really profoundly dissociative, you know, part of what happens in all sorts of psychedelics, like ketamine. And here was a patient who just experiences this. Now, it happened to be the case that for pre-surgical mapping, this person had a lot of electrodes in their brain. So we could see what was happening in large parts of the brain when they were having these seizure auras. And sure enough, when they had these auras, there was this sinusoidal activity in the retrospenial cortex, this really sine wave activity showing up in these parts of the brain, with the other parts of the brain not that affected, with the caveat that a lot of the kind of parts of the default mode network just were not sampled. A lot of it is eloquent cortex, so we tend not to stick a lot of electrodes in there in humans. Although notably, the hippocampus was not that affected, which has sometimes been implicated in the default mode network. But it's kind of fascinating that this person's kind of natural seizure aura experience of dissociation looks pretty darn similar to what we were seeing in the mice. And the last thing they did was actually stimulate electrodes in this human subject in different parts of the brain. And they did exactly that 1 to 3 hertz kind of sine wave activity, right? And in other parts of the brain, really didn't have that much of effect. But when they did it in this particular part of the default mode network, the retrospenial cortex, we were basically, or not we, this excellent team was able to recreate these dissociative experiences. So I'll just read some of this feeling of disconnected, something a little bit pleasant. It's like being weightless in your own mind as a personality. Maybe the same way a pilot can lose control of a plane, like they can be forced out of the cockpit or not control it, but still see what's happening to the whole plane. That's kind of just what happened. I got pulled out of the pilot's chair, but I could still see the gauges. You can see the information flowing. You can't control it, but you can see it, which again, I think is a pretty beautiful description of a dissociative experience. You know, I think a lot of meditation coaches would like kill to get you to kind of experience that. And so you can see that that emerges here from a manipulation of the default mode network. I threw this in at the last minute while Gianni was talking. So this is a complete pivot. I'm talking about something else now. But there has been a lot of Fmri studies kind of implicating correlations in the default mode network, especially whether the back of it here in the retrospenial cortex is correlated with the front of it in the medial prefrontal cortex in psychedelics. And people are saying, well, when you are on a psychedelic, this correlation goes away, and that could be what's causing this experience. And that could be true. But I just want to inject one bit of caution to that. So just to refresh, right? So we know this is an Fmri signal caused by nuclear resonance, ratio of deoxyhemoglobin. Turns out that depends on the head being in the same place. And what's well known in the field is that when you have head movement, a lot of it is kind of along this axis, right? Where you move your head forward and backwards while you're laying in this very uncomfortable scanner. And so if you compare somebody who's just laying still to somebody who's moving, the biggest effect you find is a loss of correlation between the front and the back of the brain, which makes sense. You're moving these around, those parts of the brain are moving, and they become less correlated because of the physical corruption of your signal. Now, if you go through the kind of literature on LSD and psilocybin and fMRI, there's actually an interesting test a lot of them do where they ask people, did you know you were on the drug or did you think about placebo? 100% of people get it right. And it's because you're having an experience, and it's actually quite difficult to be in that scanner, pretty claustrophobic and hold still when you're under the influence of these substances. So you see, some pretty dramatic changes in motion. Now, a lot of these studies have done some work to try and correct that, but I would just inject a little bit of caution that although I think there's much to be learned here, and I clearly think that there's a role in understanding default mode network activity for these experiences, that this particular finding is kind of subject to being corrupted through an artifact of the experiment. So that's basically my talk. Hopefully I answered these questions. So just as a recap, what's the default mode network? It's a set of areas in the brain who, generally speaking, become less active when you're engaged in an externally directed task compared to when you're kind of introspecting or using your egocentric model of the world, present in all species, and it tends to be correlated. These parts of the brain go up and down together. The functional piece, I think I just said, is a bit more of your kind of egocentric, your model of the world. And I think Alzheimer's kind of dramatically kind of shows you that in the different phases of the disease. And I think what specifically its role is in psychedelic experiences is still to be understood. But I think that some of the work that came out of Isaac and Sam's paper, I think pretty convincingly shows that this rhythm in this part of the default mode network that we were able to map out in mice and humans clearly was associated both causally and observationally with pieces of psychedelic experience. So that's my talk, and I'd love to hear any discussion. And Mark, you can correct the pieces that I got wrong here.
I fully enjoyed your talk. I even learned a few things. No, great job.
Thanks so much, Anish. I'm gonna just to lead off the question period, and I won't hog it. I have a lot of questions, but I'm gonna try to summarize what I feel is this layperson, this narrative about the default mode network in psychedelics. And so I'm gonna give this the summary, and then I want you to disabuse me of the things of these notions that I have wrong. So the story goes something like this, that, and again, I totally don't believe this. This is like not me, this is a friend. No, but the idea is the default mode network is correlated with, it's increased in depression and anxiety. It has something to do with this self-referential, calcified way of thinking. That's what, that's what being trapped in depression and anxiety is. Psychedelics come along as this magic bullet, and magically they loosen the connections in this network. And this maps one-to-one onto the idea of new ways of thinking, openness, psychological flexibility. So you do the psychedelic, it's this plasticity generating window, and then the networks solidify back into a resting state that is less calcified, thus less depressed, less ruminative, etc. So obviously super simplified, but that seems to be the narrative that's sort of peddled around. Certainly not by scientists in the primary literature, but I was just hoping from what you know, and Mark, please feel free to jump in too, or anybody actually, Boris is on the call, what pieces of that story do we know not to be true versus what is there some reason to believe might be true?
I've done a lot of talking, so Mark, I'd love to hear your take on this.
Yeah, no, I think you have a reasonable idea that the one of the things that's very interesting is beyond what you mentioned, that how it's used in a psychiatric realm. And one of the most startling things that kind of resurrected the psychedelic story and made it more legitimate was its use in patients, terminal cancer patients with intractable pain. And as my understanding of it is that it wasn't so much that the pain disappeared, but the tolerance to the experience was tolerable. And you may recall that there was a surgical approach to this problem many years ago, where you got in there and you cut some tracts in the brain. And I actually was thinking about this recently in the context of the anatomy of the default mode network. And basically what you did was to cut the tract between the front and the back. Singulotomy was, I think that was the name of the thing. But if you look at the connection, so this, yeah, I think this idea that you are able to stand back from yourself and kind of take a different view of it, as weird as that sounds, it kind of captures a sense of it. So I think there are examples like that. I was caused recently, I have an internist here in Seattle, and his major interest is in Alzheimer's, but I don't think I have it yet. But I had my recent visit with him, and he was noting certain things about the behavior of patients with early Alzheimer's disease that he thought was puzzling. For example, being able to plan for the future, if you will, and things like this, because you're so, when you see these people, and it's rather frequent in my family, you're so taken by the memory problem that you just kind of overlook what else is likely going on here. And yet, as Anish so beautifully showed, you're attacking this system that is eminently involved in all these high-level things. And so we have a nice conversation going, or I do, with this fellow in Seattle, as it's time to think more broadly when you cause problems like this in that system. So I think it's extremely interesting. I think the patient that was just mentioned here, that Anish mentioned, Joseph Parvizi's patient, was the most revealing sense of what it means to, if you will, lose yourself. I've just had a hard time, Anish and I both had a fine time meeting Michael Pollan, who wrote the book that's caused all this stir. And it's a marvelous book, but he even talks about losing himself on psilocybin. And I just couldn't relate to what does that feel like? And yet you look at Joseph's patient who's having seizures in his retrospenial cortex, I think you get a picture of what it means. Anyway, I thoroughly enjoyed that, Anish.
Thank you.
That was a great talk.
My sense on your question, Gianni, is that very well could be true, but I suspect there's a little bit more meat to those bones that we have yet to discover. I'd be really curious what, Boris, you think about this too, if you're listening. But my sense is that, to the extent that the default mode network, and especially the back end, the hippocampus and retrospenial cortex, are deeply involved in memory, and all that's come to imply, right? Spatial navigation, your view of the world and all of that. It probably goes through different states throughout the day. There's, of course, the encoding state where you're kind of having experiences that you're going to remember. But we know in sleep, you go through different, just completely different activity states, right? There's this thing called a sharp wave ripple that happens during parts of your sleep that we think travels through this network, would be my hypothesis, and plays a role in consolidating memories. And we also know that there's something just weird. I mean, there's dreams throughout sleep, but especially in REM sleep, you clearly are having experiences that are subjectively happening to you, right? Like we've all bored our partners with our dreams, but you somehow are able to know that that was a dream, right? It doesn't get encoded by your hippocampus in the same way as something that actually happened to you. So my complete Vernon Mountcastle moonshine hypothesis is that I wonder if psychedelics are putting the brain into a state where you're capturing pieces of something like a REM sleep, where your subjective experiences are not as constrained by the environment as they normally would be, but you're still consolidating those experiences as if you're awake. And it could be that the combination of those two things in this kind of artificial brain state has a beneficial effect for a patient, especially potentially under the influence of a good therapist. So my hypothesis would be that it's not merely the fact of the pharmacology, but the content of your experiences in this brain state that would have therapeutic effect. And that's a very testable hypothesis. I could be completely wrong about that. And I know Boris has something to say about that.
I'll hold off on that particular hobby horse of subjective experience and conscious awareness in psychedelics. But I actually was hoping either you or Mark could comment on a couple other things. This may be a little bit technical, but there's a paper from Hopkins recently that looking at a kappa agonist, salvinorin, and which is, I guess, it's psychedelic. It produces a very different kind of experience, and one of the takeaways from that paper is questioning whether these changes that we see in the default mode network are really specific to psychedelics. Like, is there any, this is a really cool, it's a great talk, and it really kind of brings it to life. And I can see why the lay press is so taken with this, way to explain some of these experiences. But I think it's a important research question is there any specificity? And the other one is really, I guess, more technical. The first time I ever, really learned about ketamine in an academic setting was in anesthesia, where we just worry about, it's not great for neurosurgery because it does all kinds of things like decoupling activity from blood flow. And so, then it kind of raised the question, is there are a lot of studies from Imperial, in London, where they're giving these drugs that are probably vasoactive and taking all these blood flow measurements. And I'm not nearly expert enough to judge, but it seems like that might be a confound.
I can answer the technical piece, Boris. I totally agree. So that's why I tried to show some evidence that was, based on calcium imaging. And then in their paper, they do electrophysiology to kind of show the sinusoidal activity. So I think that piece of it is true, but I think I am like in the camp of people who are very careful about Fmri studies, because as you're pointing out, that you do change elements of vascular physiology. Mark, you have a sense of the specificity of, in that feature? I don't really know anything about that.
This represents a story that goes back decades in terms of the relationship between brain function and its metabolism and its blood flow. This goes back to the beginning, even before the beginning of my own career. So this has been around a long time. But I think Anish, he pointed it out very nicely, that we're looking at a very interesting signal that comes out of the brain as a result of these differences between oxygen consumption and glucose utilization. That leaves the variable in there as the oxygenated hemoglobin in blood passing through the brain. But there is a metabolic side to this that I think we are only beginning to deal with. It has to do with things such as metabolic rhythms. Because you're talking here, if you're talking about the metabolism that underlies this story, there is something that if you mention it to an oncologist, they'd say, oh, sure, but it's called aerobic glycolysis. In other words, you're using glycolysis without passing it through your mitochondria. And this has all kinds of interesting implications about fueling activities in astrocytes and microglial cells and so forth. It has to do with synaptic plasticity and a variety of other things. And among other things, the default mode network is uniquely glycolytic in this regard, with a very large complement of what we call neotenous genes, which have to do with synaptic rebuilding and plasticity and the like. And the more I kind of sit at the table with this story, the more complicated it becomes over time. And so when we see these rhythms in the brain, the question is, you'd like to think, well, it's all neurophysiology. It's ions passing across membranes, it's potential differences and so forth. But one of the most well-known biological rhythms around is glycolysis. If you put a bunch of yeast in a dish and you get in there and you measure their metabolic rhythm, they all have a thing that looks very much like the trace of a Bolt signal. And if you leave it there any period of time, pretty soon they're all synced up. So they're all up and down together. And furthermore, if you leave it there a little bit longer, pretty soon they've divided up the metabolic job. They're in cost sharing mode. So you could begin to see how a biological system, a multicellular system, could go about building very elaborate relationships based on its metabolism alone. Now, this is a major diversion of where this talk was and is and should be, but just to warn you that we have a lot to learn. And I think from my perspective, that learning needs to occur in the neuroscience community, where when you bring up the word metabolism, it's like the coal you throw into a furnace so it produces heat. Well, yeah, that's kind of true, but it's far richer than that. So there's elements to this story, and of course, if you're changing metabolism, are you changing blood flow? Well, the answer is sometimes you do and sometimes you don't. And exactly what those relationships are and what they're telling us and how it leads to this is a rich area to be thinking about as we move forward. But so it's a great, I'd say for young people, it's a great time to be at the table.
Thank you. Yeah. I guess it's maybe can be taken also as for someone who's not a neuroscientist, can be taken as a word of caution, where there's a story and then there are certain features of the data from fMRI or otherwise that might be strung together to fit the story. But there's questions even about what those data points even mean, even in neuroscience, before they're even connected to a story. So I think it just.
I might add one thing. I think that you can get very reductionist about all this, and I was kind of sliding down that slippery slope. But I think that a lot of insight comes from things like clinical observation here. As you begin to reflect on what it's like to use these psychedelics, whether it's intractable pain or it's some psychiatric thing, and you get a result, right away you have data on your table there that's very valuable in understanding this. And it's unique because as cute and beautiful as this picture that I'm still staring at on the screen here with this stuff in the retrospenial cortex of a mouse, I haven't a clue about what's going on in his brain. And so I think that the clinical side of this, if you're observant and keep you have this kind of thinking in mind, it's a very valuable area. And even going back, when every neurologist, I'm a neurologist by training, going to the bedside with somebody with spatial hemi-neglect is one of the most dramatic things going. This arm is not theirs anymore, and they only eat half the plate of food. But when you go back and look at the anatomy of that problem, guess what? You're talking about taking out part of probably of the default mode network, and it's heavy involvement in your personal spatial organization. It's really dramatic. So that's kind of an isolated example of losing yourself. But in the case of hemi-neglect, you just have lost just a piece of it. So I think the clinical side of this is going to have a lot to say, and that's why having a dialogue back and forth, I think, is really important here.
I think for nothing else, what might be cool about psychedelic experiences is that they may be able to produce states temporarily, safely, that you don't have to have a stroke to have, or have seizures, or have an electrode in your brain and be at Stanford with Joseph Barresi to learn about and publish. So there's that. On that note, though, of clinical observations, I want to get to Wendy's question. And Wendy, if you're out there and you can hear me, feel free to pop in at any point. But noting that ketamine sometimes has been shown to cause these emergence reactions, where when the dissociative effect wanes, that the person has agitation as they're coming to, in a way. So this has been shown to happen in adults sometimes, rarely, but not in children. And she's wondering if this maps onto, in some way, the development or coherence or maturation of the default mode network in humans.
I can take a crack at it. I don't know the answer. But if I had to speculate, I'd say it's probably more about everything is weird to children, whereas adults, we kind of are freaked out by a novel experience. I'd be curious sorry to pick on you, Boris, but you're a practicing anesthesiologist. So, but I would wonder if you warned, adult patients about you might have kind of some of these perceptual disturbances, your body might not feel like your own, if you could get a diminishment of those, of that reaction.
How about just what do we know about when the default mode network matures in humans?
That's a good question. So it's present, it seems like, at birth. There's this story that that has been published by actually friends of mine, where they did have this beautiful kind of development that initially the front and the back are not correlated, and then they become correlated with time. And it was beautiful. It made so much sense. It seems like it's not true, though. It turns out that there was two confounds, one being this motion issue. People become better at holding still in the scanner as they get older. And the second was actually an issue of sleep. So when we were scanning like 2-year-olds, you could never get them to hold still. And we even scanned neonates, but they'd be asleep. And it turns out that there's a natural lessening of the correlation between the front and the back of the brain. And none of these effects are on-off effects, by the way, right? They're like, the correlations are 15% weaker, which is a huge effect in Fmri. And so it turns out that that's also true in human sleep. So when you and I go to sleep, those correlations go down. So the question, I think, is still there is developmentally, if you had an awake 2-year-old, an awake neonate, would that correlation be lower, or is it a function of the fact that they're asleep? So we don't know the answer to that. A half ago at a restaurant. So I still think this might be true, but I've gathered no evidence for this theory. But I guess I come from two perspectives. One is, I suspect that most kind of experiences that we can reach pharmacologically have a physiologic correlate, right? So I don't think that we can push the brain into kind of spaces that we never access in normal physiology, to a first approximation. And so when I think about the psychedelic experience, I'm thinking, okay, what pieces of normal physiology are being sampled here? And the idea that, you're having a perceptual experience that doesn't correlate at all with your actual perception, I think, of course, happens during dreaming. But if you think about that, right, that's a highly organized state with like no neuromodulatory changes and actually large endocrine changes throughout the body that are coordinated. But you could imagine that, and we know there's a memory function to that. I think it's very controversial what that function is, but we definitely see types of activity in the hippocampus propagating into the rest of the cortex, and vice versa, that we don't see in awake periods. So my hypothesis is that you are marrying kind of the awake state of the hippocampus, where you are kind of recording experiences as veridical and happening to you, with kind of this experience of perceptual disturbance. And it's possible that in some way that combination, it either like privileges those experiences to be like remembered as more profound, or it might be that the plasticity state of the brain itself has changed, and in a period in which your perceptions are not kind of as constrained by the environment. And so how that kind of works in terms of shaping your perception of the world is not clear, but my guess would be that the same kind of content of kind of thought, like, oh, the world is full of love, you know, as a thought here on Zoom between us would carry less kind of relevance than one that was arrived at with these like perceptual disturbances in different state of the cortex. But I don't know. I need to clearly think about this more.
I remember the part that I was hoping to get to at that preamble was essentially the idea that you know, you might this idea of the entropic brain, right, which is one of Carhart Harris's ideas, which mixes this idea of metaphor, you know, with randomness and uncertainty with physics principles. So uncertainty or entropy in the brain might be a mathematical idea. You can measure this, it's an empirical thing. And then uncertainty and this idea of not being constrained can also be a psychological phenomena. So in the psychedelic state, you know, or Freud's primary process, you might have looser associations, you might have this mystical experience, you might feel all connected, boundaries are loosened, et cetera. So there's it's less constrained. But to your point, that maybe even though less constrained, the experiences might still be consolidated in memory with, you know, your consolidation process is online and working appropriately. So you can have experiences that are big and wide and unconstrained, but they still stick in a sense, like normal experiences would. Or to your point, maybe not only that, but they have greater salience. So maybe they stick more in memory. That's what I was trying to say.
That was very well put, Johnny. Much better than I was rambling on about.
I'll get to, I hope I'm not mispronouncing this, but Hadi Zaki, I'm just reading from the chat and jump in if you'd like. Isaac Calvar's work indicates LSD's actually doesn't produce the same phenomena in the retrospenial cortex. Given the therapeutic potential of LSD, what does this say about the default mode network and psychedelics globally? I don't know if Hadi, you want to elaborate on those specific LSD retrospenial effects.
Yeah, no, he's absolutely right. Thanks for that question. I had to like cover up the, you know, because the movie shows all of the drugs at once. I covered up the rest because it does not produce that 1 to 3 hertz oscillation in the default mode network. If I had to guess, my guess is that there are multiple pathways to modifying the function of the default mode network, which can lead to the common outcome of pieces of psychedelic experience, which itself is very broad, right? My suspicion is that the specifics of what people tend to experience on ketamine is different than what they tend to experience on serotonin 2A agonists. And so there's other folks who have done, I think, and are continuing to do, I'm aware of some unpublished work showing different types of modifications of default mode network activity under LSD, but you're absolutely right. I think that the retrospenial oscillation is perhaps only one example.
And then Gabe, I think, joined a little bit late. To your question, Gabe's asking about, do Alzheimer's patients have reduced default mode network activity? Just as a recap, Anish was showing slides that if you map the location of the plaques that form in Alzheimer's brains, that looks very similar to when you map nodes of the default network. And so this maybe also correlates with these early symptoms of Alzheimer's, which is loss of orientation of self in context, like getting lost in a familiar environment or misinterpreting who a family member is, for instance. To your second question, Gabe, maybe you can speak to this, Anish or Mark or anybody, which is, do we see these enduring effects, for people who have had enduring positive effects of psychedelics, do we also see an enduring change in their default mode network activity?
I don't know the answer to that.
Yeah, I don't know the answer to that
Yeah,
either.
neither do I. I have a question that maybe Anish can help me with here, and that has to do with the effect of Alzheimer's disease, which is pretty devastating because the target is the default mode network for the most part, certainly at the beginning of the disease. And if I recall correctly, among the many papers that have talked about this, is that these anticorrelations between the so-called task positive and task negative collection of areas, and the task negative being the default mode network, is fundamentally lost. So you're not, if you will, continually going from one state to another as you go through your daily life, no matter what you're doing. And that surely ought to have a profound effect on your, if you will, sense of self under these circumstances. I think that was Matt Breyer made quite a point. He was another MD, PhD in the lab, that it was profound. This relationship was profoundly disturbed.
Yeah, that's a really good point. And I don't think I made this point well enough, but the talk is about this network. We spent a lot of time talking about it. But I think that, like, for me, the key kind of observation is this slide that we're looking at now, right? That there's these two giant networks in the brain that coordinate, but are mutually kind of inhibitory in a sense, right? So there's these parts of the brain that are really much more engaged when you're really focusing on the outside world. And then there's the default mode network, which is more engaged when you're kind of working from your internal model. And so I think that even more than just saying, oh, there's like a change in the default mode network, I think that just changing the balance and activity between these two networks and the coordination in between them itself can lead to kind of profound changes in how you experience the world. And I think that probably circles back to the question the other person asked about LSD, that if 18,000 citations notwithstanding, I don't think Mark or I think that the default mode network is the end all be all here, but it's kind of a principle. But yeah, like how it coordinates with other networks and this kind of idea of ego versus allocentric balance, I think is more important than the network itself.
I would add one thing to this story, the anticorrelation story, and that is performance variability, because this goes back to George Bishop in 1933, where he was the first person to talk about this. But it's a well-studied phenomenon that if you ask somebody to perform some kind of a repetitive task, that there is variability in their performance that has the rhythmicity of exactly what you're looking at here. And so that, and this fits with this, these ideas of the states of the hippocampus, where you're going from the theta state to the ripple state and back and forth. And you often think of it in terms of being asleep in slow wave sleep or being awake, but you're doing this rather routinely all the time. And it's been well demonstrated in rodents and other animals. But what you're looking at here is a kind of a two states of the system that are normally, you're actually moving from one to the other as we're sitting here talking. And you're kind of unaware of it, but then when you start maybe introspecting just a bit, and you find your mind wandering as somebody on this Zoom says something and your mind floats off in another direction, well, you're on that curve. Yeah, this was a very controversial observation when it was put out there, but I think it's one of the most insightful ones that have been made over time.
Speaking of mind wandering, and sorry, Anish, I also realized I said I'd let you go in an hour and we're already past that. So I want to get to, real quick for Lupita's question, if there's any research been done on microdosing and the default mode network. None that I know of. I don't think there has been any, but I'll check it out.
I think it's a good idea because you could potentially get around some of the confounds of motion from kind of disinhibition by reducing the dose.
That's a good point. But I just wanted to give Ryan joined late, Ryan Ash, one of our other MD PhD neuroscientist genius residents here. So Ryan, I know I see you there, so I'll just if you want to go ahead and ask your question about meditation, I think that was the link.
Two questions I had. Yeah, one is, what are like the salient differences in structure and function of the default node network between humans and non-human primates? And I'm thinking about that from this lens of, the mind wandering network, because do non-human primates, do their minds wander? And I mean, I think that, there is. Yeah, so I wonder we can look at that. And then what do you think about the data showing that meditators have decreased default mode network activity because maybe they're like getting lost in thought or lost in the self-concept less? Is that even a reasonable way of thinking about this?
Mark, I'll defer to you on these. I think you have better answers.
I was sitting here poised to hear what you had to say. And my mind was wandering. Yeah, if you go back to Yuri Basaki, who probably knows more about sharp wave ripples and the states of the hippocampus than anybody alive, and he shows these little these tracings where they're recording the state of the hippocampus as a mouse runs around and does its thing, and then it sits back and flicks its whiskers around, and it goes into these sharp wave ripple states where it's now reversed its conversation with the cortex. And you kind of wonder in your mind's eye, what is that little critter thinking about? Is there I find it fascinating to kind of think about it, but I haven't a clue.
Yeah, Ryan, I think from my take, the question of homology across species is fascinating. So pieces of the brain, like the retrospenial cortex and hippocampus, as you know, are ancient. So those are pretty well mapped between all the mammalian species that have been studied. But the parietal areas change quite a lot. And so in the monkey, the kind of parts of the parietal that are involved are kind of verging on more motor areas or areas that are involved in motor planning. And then that's even more so in rodents. And what the implications of that are in terms of how this network is instantiated differently across species, I don't know, but I think it's really interesting. And there's some work on medial prefrontal cortex and how that changes across species too, Mark, right? I think you know that story.
Yeah,
really
well, this is one of the interesting things of this slide that's up in front of us right now, but those lateral areas in the monkey, there's 4/5.
Yeah. In
the human, that's the old Brodmann area 39. And the monkey does not have a Brodmann area 39. It whatever is there, and I don't recall specifically, but then if you go to the stuff on the left and the rat, and we had some wonderful input from the anatomy people at Washington University, particularly Joel Price. And when you get there and you get into this, what there is of the lateral parietal thing in the rodent, you're now into sensory motor areas.
That's right, yeah.
So the question is, how do we move across these maps? I think you kind of pointed it out, Anish. How did this thing evolve so that when you get over to the human, you still have this same architecture, but it's instantiated in a very different, if you will, functional areas that have become refined. And what led to that? How do we think about it?
Thank you for that. Okay, I think I guess my take is that it's still a really open, interesting question. So we can keep talking and discussing.
I think there's more questions than there are answers in this business right now, but it's a fascinating business.
Thank you.
Thanks, Ryan. And I'll make this the last one. And it seems like it always comes back to consciousness. But Andrew, was this idea of similar but slightly different maps among mammals, might that suggest in some way that consciousness is a continuum rather than some sort of binary? Of course, no one has the answer, but be curious to hear your take on that, Anish.
It's so funny. I was listening into a neuroscience talk this morning, and I feel like every talk ends on consciousness.
Yeah, right.
Every career is
Exactly. And Mark's sense is like, that Mark's joke is like, that a scientist are in emeritus status when they start talking about consciousness. not to diss any consciousness researchers. I think that, my only kind of quasi-intelligent thought about this actually is it makes me think more about what Ryan was suggesting too, right? Ryan was asking, like, is there mind wandering in a mouse? And what we do know, right, is that, actually, I don't know that this has been published, but I've definitely seen evidence that this kind of idea, right, get to it. The idea that there's kind of these two broad systems in the brain, the ego and the allocentric systems, and they kind of work in concert, but in opposition. So one being kind of more active in the allocentric kind of moments of your life, and one being more active in the egocentric moments. I've definitely seen evidence of that in a mouse. So the idea that there are kind of this interplay and that there is a sense of like your model of the world, the mouse's self, if you want to call it that. I think that's probably true. And to the extent that there are different parts of the brain that are kind of that are instantiating the kind of internal model of the rodent versus the primate, probably does have implications about what that model emphasizes, and what it kind of produces and how it looks. What that means for consciousness, I'm going to defer for several decades after someone gives me tenure.
Well, I appreciate you speculating. And we'll close it there. Thank you so much, Anish, for taking the time to do this, and thank you, Mark, as well. That's a great treat, I think, to have you here. I did not know that was actually going to happen the whole time. But thank you both so much. That's awesome. And we may call on you again when more data comes out about the default mode in psychedelic work.
Thanks so much for having me, Johnny. It was nice to kind of take my rage from my day one of CL and fully flush it out.
Yeah, no, it was thoroughly enjoyable. And it always leads me to rethink how I think about some of this stuff, to hear people's questions. I'll sleep on it tonight.
Okay, well, thanks everyone for joining, and take care. Have a good night.
All righty, bye-bye.